Good afternoon. My name is Dr. Dan Simon. I'm the Chief Clinical and Scientific Officer of University Hospitals, and it's a pleasure today to join our friends again with my dear colleague, Dr. Robert Salata, the Physician-in-Chief of University Hospitals, our infectious disease expert, and the Medical Director of the Roe Green Center for Travel Medicine and Global Health. It's great to be with you again, Bob.
Thanks, Dan.
Well, what I'd like to do, as usual, before we get started into questions, is to make sure that we can give all of our friends a great update on where we are and potentially where we're going. And I come to you today with obviously significant concerns about the next 2 to 4 weeks. As you can see here in this plot from Johns Hopkins of United States daily COVID cases, we are in the midst of a very strong third spike with over 177,000 cases alone on Friday. That declined to about 166,000 on Saturday, but you still see that this peak and this surge is well beyond that we experienced in April and in our second peak in July. It's also really important to emphasize that these increase in cases across the country is associated with not only cases, but also hospitalizations. We had 69,455 patients hospitalized in the United States on Saturday, which was a record. 25 states hit record hospitalizations for COVID over the weekend. So although, as we've talked about, the age of patients with COVID is going down, there's no question that these cases are still leading to hospitalizations and into deaths. Now, what's happening in Ohio? Well, my flashing red light is on. We had nearly 7,685 cases on November 15th, just yesterday. And you see that there's no sign that this is plateauing off, just rising and rising before us right now. If you look at some statistics across Ohio, you see on a 21-day trend that cases have gone from 2,100 to now 7,853 yesterday. We have not seen a big spike in deaths, and we're very fortunate for that. 8 only in the last 24 hours on a 21-day average of 25. Hospitalizations are significantly a problem right now, 189 in the last 24 hours, and only several days ago, we peaked at 386. The good news is, and it's something that Bob and I will talk about a little bit later, is that in general, although there are more hospitalizations, hospital stays are shorter and fewer people are requiring ICU care or are dying than before. Remember, about 1,300 deaths a day this weekend compared to 2,200 a day back in March and April, especially in the New York and New Jersey areas. What's happening at university hospitals, and as you can see depicted on this slide, very similar to what's happening in Ohio in general. We have 223 COVID patients in the hospital today across the 12 hospital health system. 169 are in acute care, 54 are in the intensive care unit. And again, you can see just short of 100 patients in the peaks in April and in July, and now over 223. And with that, I would like to stop sharing and bring our dear friend, Bob Salata, an expert here. So Bob, pretty grim statistics, you know, pause for great concern as we enter the holidays. But I think that we have to give our friends and all the listeners a little bit of hope. And I thought maybe it would be great for you, as you did this morning on our Hicks call very early this morning, tell us about the announcement related to Moderna, because it does bring us hope.
So thanks, Dan. It seems like Mondays are times when we can provide a little excitement and hope. Last week on Monday, Pfizer reported on their preliminary interim analysis that in comparing cases of true blue COVID between vaccine and placebo recipients, they had over a 90% efficacy rate and terrific safety progress in that regard. So too, the Moderna Data Safety Monitoring Board met yesterday, and they reported this morning that in their analysis of 95 cases of COVID, there are only 5 in the vaccine recipients and 90 in those that received placebo. And that accounted for a 94.5% efficacy rate. So now we have 2 vaccines that are showing tremendous efficacy rates. In the Pfizer's case, we will see a upcoming further analysis when they reach 168 cases. Their analysis was based on 94 cases. They should get there by early next week. And if this holds up as we hope and think it will, then that's just great news. And what that means in that both of these companies will be submitting their analyses to the feds, especially the Food and Drug Administration, probably by the end of November, early December. And if they're approved for early access utilization, then we should potentially have vaccine. And we'll talk about the priority groups, et cetera, in a few minutes, to the tune of about 20 to 40 million vaccines available in the month of December. So that's great hope as we are on this rapid journey. But last thing I just want to emphasize is that for all the vaccines, and there are 2 others currently in play and being studied in the US, aside from these 2, this has moved rapidly, more so than we've ever seen before with vaccine development. But I don't like the term Operation Warp Speed that suggests maybe you're cutting corners. And I like the term that was coined by the CEO of Pfizer, saying that we're moving at the speed of science. And in that regard, these studies have been conducted incredibly carefully, and no corners have been cut at all. We could talk about the makeup of these 2 vaccines, which are similar as we get there, but really more hope added to the initial Pfizer announcement and now from Moderna.
So Bob, I think we do want to encourage all of our listeners to go to Q&A and ask your questions. And actually, a first question did pop up, and it's a very good question. And this question comes from Linda. And Linda wants to know, what exactly is the measure of efficacy of these vaccines? What do they mean by 90% or 95% effective?
Yep, great question. And that's the most critical part of this. And it will be the major point of education of the public in terms of getting them to accept the vaccine as we move along. There are the major point of efficacy or effectiveness is looking at true confirmed cases of COVID in the vaccine versus those that received placebo. And obviously we'll want to see these kinds of numbers that have been reported. So that's calculated on the basis of the total population vaccinated versus those that developed COVID-19 in both the vaccine and placebo areas. That's where these numbers are derived from. The other surrogate numbers of efficacy that you've all heard about are the antibody response that we see from all of these vaccines, and they've been reported in the medical literature already. And that is, we're looking for so-called neutralizing antibody levels. These are measured in the laboratory from all of these recipients. And we do think, in the end, that sufficient amounts and the right kind of antibodies are going to be important for this protection from an immune response point of view. The other thing is there's another arm of immunity called T cell responses, and those have also been reported to be vigorous. Let me just end by saying in the antibody responses by both companies, they've published and reported that these are anywhere from 10 to 20 to higher responses than what we've seen with natural infection. And we'll be measuring these antibody levels over time to see how long they're sustained. That's an unanswered question at the moment.
Terrific, Bob. Well, we're getting, the questions are pouring in, and of course, I made a couple slides to help us answer some of these questions in anticipation. Right. And so what I'd like to do is share this slide, and this addresses some questions by both Linda, Debbie, and others. And that is when I'm terming vaccine facts, something about vaccine efficacy, and then a question about the storage issues. So the first, I think, which is really important, this comes from the Centers for Disease Control, as you can see right here, is that this is looking at the effectiveness of vaccines that we know a fair bit about, and then we can look at the effectiveness of the COVID vaccine. So here you see tetanus, rubella up in the 95% range, and then lower effectiveness, as we all know, from the flu vaccine, which varies from between 40 and 60%. So at 90 to 95%, we have a vaccine, Bob, wouldn't you say, that looks almost as good as measles and rubella.
Absolutely. And just to remind folks, last year was also a milestone in terms of number of cases and deaths related to measles. And that was in part related to the fact that there were many parents that declined to have their children vaccinated. But the important point here in the whole arena of infectious diseases is when we have an effective vaccine, that is the best measure that we can provide. And so too, this will be the case with these numbers if they hold up, and we think they will, for COVID-19.
Now, the second panel, Bob, entitled New Lows, addresses the concern that several questioners have related to the practicality of the Pfizer vaccine because of the fact of its need for very low temperatures as a messenger RNA vaccine, which can be less stable. So let's just walk through this quickly, and then, Bob, I'll let you talk a little bit about distribution. This is a temperature scale in Celsius, 0 at the top and minus 80 Celsius down here. These are dry ice temperatures. So if you were to ship ice cream or frozen steaks like we sometimes do over the holidays, that requires dry ice in the minus 80 degrees Celsius range. Here you see other common vaccines, the flu vaccine, 0 to minus 10, chickenpox, measles, mumps, and rubella. These are really regular freezer type temperatures. And then, Bob, I'll end with you, and then and then have you take over. AstraZeneca and J&J are really refrigerator level temperatures, Moderna freezer, and then Pfizer, as we say, a minus 80 or dry ice. How does that figure in, Bob, into how we're going to get this vaccine out and how people are going to get vaccinated?
Right. Great question. And let me take it from here. There was a New York Times article recently that addressed the storage and logistical issues related to these vaccines, particularly Pfizer. And actually, I was on over the weekend on CNN and discussed this very issue. So these temperatures that you mentioned are what is at the South Pole. That's very, very cold. We don't want to go there, right? But what Pfizer has devised at this time is a special storage pack, if you will. It's like a cooler, into which each of these vaccines, which have 5 doses per each of these vials, are packed, and then dry ice is placed on top of this. Now, with that said, once transported to hospitals, clinics, pharmacies, even the retail pharmacies, which many have been enlisted as sites for vaccination by the feds, these can be kept in the regular freezer compartment, given that special storage unit and also the dry ice, as long as it's replenished every 3 or 4 days for 15 days. So I think that is more encouraging rather than thinking that physicians' offices or retail pharmacies have to purchase these very special freezers that will allow storage in a -70 to -80 storage situation. So it's not quite as bad as we thought. And they are going to provide this, widely. So that is the storage containers to provide that possibility. Keeping things in the freezer is a much more viable option than to expect this to happen under the current circumstances, which was very controlled, as you know, at our Pfizer study in that type of storage capacity. Now, Pfizer is also looking at lowering the temperature requirements so that they're looking even at -20 and even refrigerated temperatures to see if that will be sufficient. Because after all, the Moderna vaccine is very similar in its makeup to the Pfizer vaccine. That is, it is with messenger RNA, and I think you do want to show that later, Dan, coupled to the spike protein of this virus. Many of you have seen pictures of the virus, and these little projections out from the surface are indeed, there you go, what is called the spike protein. And the other thing that both of these vaccines demonstrate thereafter is that this was the target for the immune response to these vaccines. And this is based in part on prior experience with SARS-1 in the early 2000s, and another one called MERS, which arose from the Middle East, another coronavirus. Both of them were that developing antibody responses against that spike protein shown in the right lower panel is really very important in those days and against those viruses in providing protection against continued infection or reinfection. So that verifies and codifies the fact that that was the right choice in terms of what to couple this messenger RNA to. And I think that's very important that, we think that that is really the key in terms of moving these vaccines forward.
So Bob, just to review, the genetic vaccines, which either have DNA or RNA, and in this case, both the Pfizer and Moderna are RNA vaccines, right, fall into this category. The viral vector vaccines would be both Johnson & Johnson and the AstraZeneca Oxford vaccines. And one of the questions, Bob, that's coming up, obviously both of them are sort of this result here, which is that here is the COVID-19 virus, and we're trying to make antibodies that block the spike protein. The spike protein is the way the virus gains entrance to your cells, both in the nasal passages and in the lung, and that's how it causes the damage. So these antibodies block the entrance of the virus. So one of the questions, Bob, that people have, of course, is which vaccine should I get? Obviously, in the beginning, it appears that we'll have 2 choices from messenger RNA vaccines. Then down the line, AstraZeneca and Johnson & Johnson are modified adenoviruses. And I guess the question would be, is one better than the other? Is one safer from the other, or do we not know yet?
We don't know yet, but I would just say this, that In the AstraZeneca vaccine, there were reported side effects. Very early in their studies, there was a condition called transverse myelitis, which is inflammation of the spinal cord, which can result in weakness of the arms and legs, particularly the legs. And sometimes that can be partial and reversible, but sometimes not. And in that regard, it was felt that in the early case, and this has not been as well publicized, that that person had developed very good evidence clinically and by laboratories of having multiple sclerosis, the very first presentation of that, which is one of the other causes of that condition. And there are several others. And the second example where this occurred in their phase 3 testing, that is the last stage before it's made available for use in the public, this condition occurred again. I had reviewed, and I've spoken before, about the fact that in the US, there is a database kept by the Food and Drug Administration on all side effects to vaccines. And over the last 15 years, there were 119 cases of transverse myelitis reported, 15 years. But many of those occurred well within over 6 to 12 and even later. And it's hard to establish a cause and effect relationship. When it occurs earlier, after the vaccine is administered, then of course that comes about. In the AstraZeneca situation, there were independent review boards that looked at this and reported to their FDA equivalent in Great Britain their findings. And they felt, even though this occurred in a vaccine recipient, they couldn't establish firmly a cause and effect relationship. So we have now, in turn, reviewed this information at the FDA level and the National Institutes of Health, because they are joining forces with AstraZeneca and pushing out this vaccine. There have been no more of these types of conditions reported. There was a death in Brazil with AstraZeneca, but that appeared to be in a non-vaccine recipient, actually a physician who developed COVID and died of complications thereof. So always we have to scrutinize these side effects very, very carefully. I would only point out that as of now, we have not seen this in either the Pfizer or Moderna vaccines. But remember that one of the things we're doing in all of these studies is following folks up to 24 months after the vaccine is initiated. And that is to be absolutely certain of no late occurring side effects that may be seen. And the public has to be reassured about not only efficacy that we just spoke about, but also the safety of these vaccines as we move forward. So I don't think we'll ever have, Dan, at least in the short run, any comparisons between vaccines. It's just not going to happen. We can look at things like the efficacy rate of Moderna versus Pfizer, and I would say basically they're about the same. And the side effect profile in terms of what one individually chooses to receive. I would also say that it would not be something we would mix vaccines. You get a Pfizer vaccine initially, and then the second one would be a AstraZeneca vaccine, because it's just not been studied that way.
So Bob, a lot of questions about booster, no booster dose, et cetera. So we know Johnson & Johnson is a single shot,
Right.
very stable in a refrigerator. So ideally suited perhaps for global use and developing countries. The Pfizer vaccine is a booster at 21 days. When is the Moderna booster? 14 or 21?
It's 14.
14 days. So second shot either at 14 days or at 21 days. Now, I'm going to go back on sharing the screen because very good question from several people about who gets the vaccine first. When is it going to be available? And so what I'd like to do is share with you the COVID-19 vaccination program guidelines from the CDC and the FDA. We have shown these before, but I think it's really important to review them again. Phase 1 is when there are only limited doses of vaccine available. And our best estimate is that that will be now in December of 2020 when vaccine will be available. And that will be distributed on a phase 1A phase 1B approach. Phase 1A are first responders and frontline healthcare workers who are at risk of either direct or indirect exposure to patients or infectious materials. Phase 1B will be other healthcare workers, and then people at high risk of severe COVID-19 illness, especially people over 65 years of age and older. And then that'll very rapidly turn into phase 2, where a larger number of doses will be available. And this will include, of course, wider definition of healthcare workers who may not be frontline, and then critical populations defined as risk factors of heart failure, lung disease, active cancer treatment, and immunosuppression. I think it's really important to point out that just recently, really in the last 24 to 48 hours, we have received notice that the first distribution of vaccine to the state of Ohio will only include approximately 30,000 doses initially. Now, we have contracted for 100 million doses with Pfizer, which would be able to treat 50 million individuals, but that's obviously not going to happen all at once. So we do have a task force in the health system to start to identify what are the priority of healthcare workers, even in our own system, that would gain access to the very early doses that are available. I think if you talk, and Bob, perhaps I'll give you my best guess, and then we'll turn it over to you from your special connection to Dr. Fauci. But it does appear that we will be getting vaccine in sort of the mid to end of December and in the first quarter. But widespread vaccination of the general population is likely not to take place until the May-June timeframe. So perhaps, Bob, you can fill in a little bit more on the timing issues of the vaccine.
So thanks, Dan. First of all, Pfizer is doing another analysis right as we speak. And that is at day 14. That is 2 weeks after the vaccine was initially given, to see if that was associated with the same level of efficacy that we spoke about after 2 different injections. And obviously, if that's the case, that will mean that we could potentially get away with 1 injection only in this particular vaccine. All of the vaccines, as you mentioned, except for Johnson & Johnson, are double doses or 2 separate doses. And in that regard, that's another logistical challenge, getting people back after they've received the very first one. So that said, over the weekend, Dr. Fauci also said the cavalry is coming, to use a metaphor. And he estimates that based, especially now with Moderna's new information, that we may have vaccine available for the general public by as early as April. That's really modified from what we used to think would be the summertime to early fall. And that would be tremendous if we do have that. Of course, Dan, as we've spoken about before, it's convincing people to receive the vaccine too. Various polls have shown about 50% of people are willing, like you were in our study, and thank you for that, to take the vaccine as soon as it becomes available. 25% said they would wait to see about how this goes with others first, but would still be positively inclined to take the vaccine, but 25% say no. And I think with the anti-vaxxers who have now joined forces with the anti-maskers, that's going to be a hard population to get to. But we've got to do everything once we have this data in hand to initiate an educational campaign, not only in our system, but in the United States and globally, about why this is so important as we try to make a dent in the numbers that you're showing now.
So Bob, the questions are really flying in and really great questions here. One is about potential mutation of the COVID-19 virus, and will that adversely impact vaccination? Or are there different strains of COVID, just like how our flu vaccine has strain changes every year. Any comments on that?
Yeah, so studies have shown that this particular virus has mutated at least 40 times that we know of to date. And in fact, the current prevailing type in the United States is one that has mutated to the point where it's more contagious, if you will. But importantly, it's not necessarily associated with worse clinical outcomes. And that's an important concept. In all of the mutations reported to date, for the most part, what has not changed is that spike protein. And to that end, as I've already discussed, that's the primary focus that all of these vaccines are taking in terms of developing an important immune response. And hopefully it will be long lasting. Could it occur that we'll have to revaccinate populations in the future if the mutations occur and they affect the spike protein? That's a possibility. But good news so far is we have not seen that. And that's as opposed to the flu vaccine, where every year there may be subtle or sometimes major changes in the outside covering of the flu vaccine. And to that end, vaccines that contain past types, and we make our best guess every year as to what we're going to see in the United States based on what happens in Asia and in the Southern Hemisphere. Sometimes we're wrong. But I would just use this as another opportunity to tell our donors and other important folks that getting your flu vaccine is still important. Even though the Southern Hemisphere saw little in the way of flu infections, in part because of all the infection control measures that we put into place, Americans behave differently. And so we could be faced with what is called the twindemic coming up. That is both COVID as well as influenza, which have overlapping symptoms, and it's very hard to distinguish. So therefore, we're going to be testing at university hospitals in the near future for both in a single test. But having said that, even though the efficacy rate that you showed before for flu may be as low as 40 to 50%, Dan, having breakthrough influenza despite getting a vaccine is still associated, especially in older folks, with more or milder disease, less hospitalizations, and death related to influenza and its complications. So I think that's an important concept. Hopefully, this will not evolve in the same way that the flu vaccine, the flu virus does. And so just stay tuned about this because this is always an evolving issue.
So Bob, we have a lot of very, very smart people on the phone who are asking these great questions. Here's this one, which is a good one. So tetanus, approximately every 10 years, you need a booster. My hepatitis B vaccine, I still have antibodies. That was over 20 years ago. How often do you think we're going to need a COVID vaccine? I know we're gathering that information. Will it be once in a lifetime? Will you need to get a booster after 5 years?
Hopefully it'd be once in a lifetime, but we don't know that. And by the way, I just spoke about influenza. They are working on vaccines for flu that won't have to be repeated on an annual basis as well. And we're getting closer to that via studies from the National Institutes of Health. So it all depends on the good question previously about mutation, genetic mutations in COVID or the coronavirus, and whether those affect in the end the spike protein. And if they do, I think we may have to vaccinate again in the future. I can't predict how often that would be because we're still in the midst of evaluating this. And as I said, for the Pfizer vaccine and the others, we're going to be studying individuals for years afterwards, looking at whether their antibody responses and T cell responses wane. And we'll continue during that 2-year period to also look at cases of COVID in the vaccine versus placebo recipients. So I wish I had a crystal ball to tell you that it's once in a lifetime, but we don't know that right now.
So another question, Bob, has come up about the issue of mandatory vaccination. One, maybe possibly to go to school, just like we have vaccination schedules that you need for school. Pfizer obviously is vaccinating 12 to 17-year-olds, so we are going to learn about kids. What about places like university hospitals? You're going to have to play a role in determining, will we have mandatory vaccination required for healthcare workers?
So I think that's a great question. It's been undecided at this point. It is a national conversation as well by many expert groups as to whether or not we do this, including our own healthcare workers, as you implied. But let me remind you of what happened at university hospitals when we did not mandate influenza vaccine and what happened after we did. So that's an example of where that came into play. Our rates of vaccination among our healthcare workers was around 70% at best. And when we mandated this last year, for instance, we jumped well over 90%. And in that case, there is very good evidence that by getting vaccinated among our healthcare workers, just as this information I just imparted applies, we also protect our patients. There's less absenteeism among healthcare workers. There's less transmission in family units. So I think that those would be similar arguments that have been made for vaccination of school children, university-based young folks, and also for healthcare workers and first responders. Otherwise, it will be a voluntary process, but we don't know whether we're going to move in the direction of making this mandatory. That may be a decision, like we did with flu, that individual institutions will have to make that decision in the end, although there'll be recommendations coming forth from other places and authoritative agencies.
Terrific, Bob. Now, I guess another really important question is, we have seen a record of hospitalization. 69,455 were hospitalized on Saturday. Interestingly, deaths are 1,300, which is obviously taking our total US death rate to over 246,000. It's just a staggering number. But people have pointed out that the death rate is going down amongst hospitalized patients and that hospital stays are shorter. Could you tell us a little bit about why you think that's happening? I mean, that obviously is good news. We do have new treatments. Could you review those treatments and
Sure.
also tell us that, you know, your course will be better now than it was in March and April?
So this goes back first to who's getting infected these days more frequently. And that is, we're about 1.5 to 2 decades less in terms of age than we used to see at the beginning of this pandemic. And therefore, these folks generally have less underlying diseases that place them at risk for complications and death. That's not to say we haven't seen it in younger age groups, but that's less so. But the important message we're trying to get out to younger people is that they can become carriers, if you will, and a reservoir for infection to our aged and those with vulnerable conditions. And actually, the pendulum is swinging a little bit now from those very younger age groups, a little bit more to older folks again, not quite to the level we saw before. So that's part of the issue why death rates have not occurred as frequently as before. Secondly, death rates in general lag behind total cases. But having said that, I think it is true because it's held up steadily, and that's been pretty flat compared to the cases going up exponentially, as you showed. And I think that we have better treatments. We know how to care for these patients better. I mean, just yesterday, as you mentioned, with over 220 folks in our system that are in the hospital, we actually approved about 24 requests for remdesivir. As many of you have known and read, this is the only FDA proven or approved antiviral drug that we have for treatment. But it's not a panacea either. In many studies, particularly those conducted in the US, It has decreased length of stay, which is an important issue for them not to go on to develop other hospital-associated conditions and die from those as well. So I think that's laudable. And in one study, where a randomized controlled trial, which is kind of the best we can do by the NIH, published in the New England Journal of Medicine several months ago, there was a tendency towards decreased mortality in those that received remdesivir. But we need to study this drug earlier on, not just with moderate to severe disease. Can it prevent those complications from occurring? And can we give it in shorter time periods? For instance, right now, we're giving it for 5 days intravenously in hospitalized patients. And that in some patients who have turned around pretty quickly, that delays their discharge as well. We're in the process as an institution to look at whether we can do some of this in the home care setting. More to come on that. Gilead, who manufactures remdesivir, is looking at inhaled treatment, which makes sense because this starts in the respiratory tract, as well as a 3 versus 5 day course of therapy. But it's still intravenous. We need antivirals that are orally available, like Tamiflu for influenza. And we need also the big push now, because this is really a biphasic illness, COVID. It starts with viral infection that replicates and disseminates throughout the body, some to the brain, heart, kidneys, for example. And then there's this vigorous second phase where people have this robust immune response. So many studies are now looking at medications that can mitigate that robust response and are being combined with also remdesivir to see whether or not that's additionally helpful. So I think that, plus many of you have heard recently about EUA approval by the FDA for a Eli Lilly monoclonal antibody, but it did not work in patients already hospitalized. And that the message there is that if it's going to work, and we have an active study looking at this now, and since the EUA occurred, early use authorization, we are getting some of that by tomorrow here at university hospitals. So to use this earlier in the course, and many of you know that President Trump received such a cocktail initially, and as well, maybe even in a preventative way for folks that were very heavily exposed. Could this also be given to promote antibody responses and therefore abort the development of disease? Remains to be seen, but I think that offers some hope in that regard. Lastly, our colleagues from the University of Oxford and the UK not only were the inventors of the AstraZeneca vaccine, but they also conducted a study in the UK of using a very strong steroid called dexamethasone. And when they applied it to individuals who had some degree of lowered oxygen and the need for oxygen supplementation, they found a 35% decrease in death rates. And I think that's an important statement as well. So the use of steroids like this have now become standard of care. Remdesivir as well. And we're managing that through an adjudication committee because there's still limited supply of this right now. And generally speaking, we have not turned down many of these requests because they're all legitimate. So that's on the treatment front. So we need new antivirals. We need more drugs that will mitigate the severe immune response. And that's where things stand right now. The other thing that came into play, and one study is still going on here, is the use of so-called convalescent plasma. That is, individuals who have recovered from COVID, we take blood from them and take the plasma component, which has antibodies in it that they've developed against COVID, and give that to another individual, like we do blood transfusions. And so far, the jury is out on that, but that's not been a tremendously successful strategy, although it's been useful in other infectious diseases before. Ebola, for instance, where it was tried. And the only group that really benefited were folks not on a ventilator, were younger, and also in their plasma that they received, there were very high titers of the antibodies. So that means to me that the approach using monoclonal antibodies, which are very high titer, really is a viable one that we need to play out in these studies. So that's where we stand with regard to treatment right now.
Great. So Bob, we're going to shift gears now because everybody has questions about the holidays. And this is obviously a really, really sensitive issue. It's the concept of what we call bubble expansion. What happens when you take a family unit, father and a mother, perhaps one kid in high school, and then all of a sudden the rest of the family comes piling in for Thanksgiving, bringing with them all of the exposures of their kids or grandkids into the family. And with asymptomatic spread being a problem in the younger ages, this poses some challenges. So I guess, Bob, the first question I have for you is, aside from the importance of physical distancing, masking, and hand washing, what else can we do? Is there a role, for instance, in testing? That's the biggest issue.
Yeah.
And asymptomatic testing, which is very difficult to come by in the US, especially in Northeast Ohio. So tell us a little bit about Thanksgiving. What are you doing in your family? What do you recommend?
So we're not having a large family gathering. It's going to be restricted to our nuclear family. And if you look at some of the recent cases, I think it's important to understand that is in these family gatherings that we're seeing many cases now being transmitted. And we thought that was a safe thing to do by minimizing numbers and undertaking those infection control measures. The other places where you never know what other family members are doing are gyms, eating in person at restaurants, cafes, coffee shops, and the like. Those are really part of what's driving this, in addition to poor adherence to the measures that we've talked about. And I think younger people, especially as you pointed out, and some of the real spikes at the beginning of this third phase were in university towns and associated with the students because they're less apt to pay attention to these things. They think they're invulnerable. But I mentioned this before, they serve as a reservoir for transmitting this to others. And in the age groups we're talking about, even high school and above, those are the folks that are often more asymptomatic. And up to 40% of cases that where contact tracing was done and subsequent cases were found to be positive was presumably from someone that was asymptomatic at the time they had this. And you could also be transmitting the virus during the so-called pre-symptomatic stage that may be as long as 2 to 3 days before those symptoms develop in any given individual. So it is being discouraged by many authoritative groups that Thanksgiving gatherings and subsequently for the other holidays coming up, that you try to minimize the number of people. If you're going to do this, of course, we understand why. So that's something that's been seriously offered as a consideration for you. The other thing is testing. So we don't have the capacity to test with our best method, and that is the molecular test called PCR, polymerase chain reaction. We just don't have the capacity to do that. We are restricting that primarily in our system to individuals that are symptomatic, have had exposure, and including our frontline clinical workers. Beyond that, however, there are a number of other tests that we've spoken about in the past, but just to mention, one is antigen detection. And this is detecting a piece of the virus, if you will. And the advantage is it could be done quickly. You'll have an answer in 15 to 20 minutes or so. But there are both false positive, meaning it's really negative, but they're testing positive, and false negative results. The false negative results may be as high as 20%. There's a new Abbott test that's been commandeered by the feds that shows levels of reliability that are the same as the current PCR tests, but those are not widely available yet. But once they become so, I think that will be another strategy to carefully consider. Then there's antibody testing. And right now that's all over the board. In terms of the FDA, they originally, because in their haste to have testing available for antibodies, allowed almost every company to put them up. But then some were as reliable as low as 30% or so, and that doesn't help either way. In fact, at one time, certain countries like China and Chile wanted to give so-called passport immunity, or immunity passports, if you will, to individuals in their countries that tested antibody positive. But that was, I think, quickly, countered by not only the World Health Organization, but the Centers for Disease Control, et cetera. So we don't know if those are the right antibodies either. And what we want are antibodies specifically directed against the spike protein that I spoke about before. And it was those antibodies that developed in animal models of these vaccines already published that were most important for decreasing any disease in those animals given vaccine beforehand. So you can get an antibody test, but the problems are the following. If you get antibody tested and you're asymptomatic or only have mild COVID, those are not very robust, and they may wane relatively in a short period of time. The more symptomatic you are, the higher your antibody responses are. And newer studies show that they can last now, and they're still following that out, for up to 6 to 7 months. And we hope those are the right antibodies, right, given what I said, in relationship to pushing out vaccine, et cetera. So if you're going to ask a loved one who lives in El Paso, Texas, where it's really rampant right now, to be tested, I think it's still problematic in using those tests to make a decision about allowing them to come back into the family setting, bottom line.
Yeah, so I think this is really important, and a couple of things that are really worth emphasizing. One is your viral PCR positivity depends on the time from your exposure. So if I'm exposed to someone with COVID yesterday, I'm very likely to be PCR negative today. Right. Typically, we would want to wait at least 3 and ideally up to 5 days to say you're truly, truly negative. Yeah. So what does that mean? It means that if your son or daughter gets a viral PCR test in Boston on Friday and then flies home to see you on Saturday or Sunday, it just tells you about their status on Friday. Right. It doesn't tell you about what happened when they went to the airport, flew on the plane, came home, or if, for instance, they were exposed on Wednesday and Thursday before they got their viral PCR test on Friday. So you have to be it's really important to understand that means the person's negative that day. And there are a lot of limitations. If resources were unlimited, you would go with the NFL and NBA bubble approach, which is you test everybody every day with a viral PCR. But it's just impossible to do that. We are performing somewhere between 1,700, and I think we set a record this weekend, almost 2,300 viral PCR tests a day. We test every admission, everybody undergoing surgery or a procedure, all symptomatic individuals, and all healthcare workers who've been exposed or who are in a contact tracing loop. And that really takes us almost up to our full test number. If you need to do an asymptomatic viral PCR test, we refer you to commercial providers, those available through your local pharmacies. There are some online resources that you can also use where they will send you a kit, and you send the kit back, and in as short as 24 to 48 hours, you can have a turnaround. But we just do not have the availability for widespread asymptomatic PCR testing. Well, Bob, the hour's late. It's 12:55, and I want to thank you for a tremendous review. The vaccine field obviously is taking over with the great news of both Pfizer and Moderna. We need a little catch up now on some antivirals, as you pointed out, because we are going to need both. As you've heard me say over and over, we need a Tamiflu equivalent, an oral effective antiviral, as well as a good vaccine. I want to thank everybody from IR&D who helped today. We have a whole team behind this. We have recorded this, and we'll be sending this out if you'd like to distribute this, obviously to your friends. And also, just want to remind all of you, we are hitting a tough time. I'm sorry. We are here to support all of our healthcare workers. Thank you.
Thank you, Dan. And thanks to all of you for listening in again.